Hello friends, welcome to our channel Cosmos Study Circle. Let's go start the video.
Yesterday our topic was the structural organization of cell organelles. In this respect, we are taking an example, the mitochondria, the most important cell organelle, the mitochondria. We will discuss about the mitochondria in this section. First, have a look at the historical background of mitochondria. Kolliker in the year 1850 first observed mitochondria as granular structure. Fleming in 1882 demonstrated thread-like bodies in many cells and called them philia. Altman in 1892 referred to the mitochondria as bioplasts. C. Benda in the year 1898 developed the crystal violet staining technique and the structures mitochondria. Palade and Josland 1940 to 1950 worked out the fine structure of mitochondria under the electron microscope. Let us have a look at the size, shape, number, distribution, movement, structure of mitochondria. Size. The average length of mitochondrion is 3 to 4 micron and the average diameter 0.5 to 1 micron. Shape. Mitochondria vary in shape but are generally filamentous or granular. If a filamentous mitochondrion swells at one end, it gives a club-shaped appearance. If the swollen end hollows out, then it becomes tennis racket-shaped. If there is a central clear zone, the mitochondrion becomes vesicular. Number. The number of mitochondria varies in different cell types. The number of mitochondria depends upon the metabolic activity of the cell. In a normal liver cell, there are 1000 to 1600 mitochondria, while in plant cells, their number is very small. Distribution. Ordinarily, mitochondria are evenly distributed in the cytoplasm. In columnar or prismatic cells, mitochondria are oriented parallel to the long axis of the cell. Movement. Mitochondria may move freely in some cells, carrying adenosine triphosphate, that is ATP, wherever required, or may be localized in a particular region. Movement is less in animals than in plants. Often movements are rhythmical. A structure of mitochondria, very important topic, the structure of mitochondria. The mitochondrion is bonded by two membranes, the outer membrane and the inner membrane. The space between the two membranes is called outer chamber or intermembranous space. It is filled with a variety of fluid and is 40 to 70 angstrom in width. The space bounded by inner membrane is called the inner chamber or the inner membranous space. The inner membrane space is filled with a matrix which contains dense granules, ribosomes, and mitochondrial DNA. The granule consists of insoluble inorganic salts and are believed to be the binding sites of bivalent ions like magnesium and calcium. The side of the inner membrane facing the matrix side is called the M side, while the side facing the outer chamber is called C side. 2 to 6 circular DNA molecules have been identified within mitochondria. They may be present free in the matrix or may be attached to the membrane. The enzymes of the Krebs cycle are located in the matrix. In general, the outer membrane contains more proteins and phospholipids than the inner membrane. Phosphatidylcholine is the predominant lipid of the outer membrane. The inner membrane, on the other hand, contains most of the diphosphatidylglycerol. The inner layer of mitochondria is produced into plate-like or finger-like folds, which are called cristae. On the inner layer of cristae are found small starch particles or repeating tripartite units, which are sometimes called respiratory assemblies. Each starch particle consists of a base, a stalk, if not particles, that is if you may say, and a spherical head. The spherical head consists of a soluble coupling particles called F1 particles, which is actually the enzyme ATPase. It catalyzes the formation of ATP from ADP and vice versa. The oxidative phosphorylation takes place on the start particles. Mitochondria also contain DNA and RNA. The mitochondria is also called powerhouse of the cell because huge amount of energy liberated during aerobic respiration is trapped inside the mitochondria in the form of energy-rich ATP molecules, which is then utilized in performing various metabolic processes of the cell. Their specific features also includes enzymes for heme synthesis of, for example, for cytochromes and hemoglobin, occurs in mitochondrial matrix. Inner membrane is the site of electron transport system. The ATP synthesizing complex and enzymes involved in fatty acid synthesis. Enzymes in outer membrane include those involved in oxidation of adrenaline and serotonin, and others engaged in phospholipid metabolism. The two membranes differs in their permeabilities. Outer freely permeable to a small and medium-sized molecules and ions. Inner permeable only to a small unchanged molecules such as oxygen and undissociated water, and permeable to glucose and NADH2. But with various transport proteins permitting exchange of ATP and ADP, Krebs cycle intermediates, and the accumulation of pyruvate, calcium and phosphate ions against concentration gradients, all coupled to electron transport. The ATP synthetase complex F0 F1 ATPase is also embedded in the inner membrane, comprising at least 9 different polypeptides into separate units. 5 make a spherical head F1 ATPase, which can catalyze ATP synthesis. A remainder comprise a portion of channel F0, through which protons re-enter the mitochondrion after ejection by the electron transport system. That's all about the mitochondria. Thank you very much.
साथियों, वीडियो की नोटिफिकेशन पाने के लिए चैनल को सब्सक्राइब कर ले। अगर आपको कोई और जानकारी चाहिए तो नीचे दिए गए नंबर पर कांटेक्ट कर सकते हैं। जल्द ही मिलते हैं फिर एक नई वीडियो के साथ।